many of you would believe me if I told you that there is an entire world that is invisible to us? Well, I would have to tell you that it's true, and we can see that world through microbiology. So I'm going to be telling you about the basics of a microbiology lab. Now, I teach microbiology as a lab assistant here at AWC, and for all of you who may take this class in the future or not, it's still very, very cool information to know how to see this invisible world, right? So the basics of a microbiology lab, what are they? We're going to talk about how to prepare for a lab safely and keep things sterile. We're going to talk about how to take a culture and how to examine that culture in a microscope. So, lab safety. What do we got to do? We've got our lab coat on, we've got gloves, and we've got safety goggles. And that is going to keep us safe from germs. Now we're dealing with germs, which means we want to keep the area that is not germs sterile. So I have sterilized this bench using ethanol and wiping it down with a paper towel. But there's germs in the air, so how are we keeping this sterile? Well, I've got a Bunsen burner going, which has fire, and that heat is going to keep the air around this bench nice and sterile. This is a sterile zone, is what we're calling it. Alright, now that we've got our equipment on and we've got our zone nice and sterile, how do we do a lab? So, taking a culture. What we're going to do to take a culture is we're going to use a sterile cotton swab and water. So water, sterile cotton swab. Let's say I want to sample the palm of my hand. I've washed my hands already, so the only germs we're going to find are the germs that live on me all the time. So what we're going to do here is drop a little bit of water onto my palm, which is going to make the germs nice and loose, and we're going to rub that with the cotton swab, and that's going to pick up the germs. But there's not enough germs on this cotton swab to look at in a microscope, so what do we do then? In that case, we need to culture it, right? So we're going to take an agar plate, which is basically science jello, right? So what we're going to do is put the germs onto the agar, and that is going to feed them so that they grow and so that we have enough to do experiments on. So we're going to grow that in an incubator for 24 hours. And that's just going to give us enough germs so that we can pick them up and look at and do lots of other fun things to them. Let me put on another glove. And I've already incubated a plate for you guys to look at. So here is a plate from earlier with my palm germs. So I've labeled the plate properly with my initials, with where I got the sample from, the date I took it, and the temperature that it incubated at. If you notice, that is body temperature. So you can see these germs are all of the little spots. So those are like little cities of bacteria where they've grown and they're flourishing. So how do we examine these in a microscope? What we would do is we would pick up the germs and put them onto a microscope slide. That's a complicated process and it takes a really long time, so we're not going to do that. I'm just going to show you how you would use a microscope. So, if you've never seen a microscope before, this is what they look like. So there are objective lenses, which are ocular lenses, which are for your eyes. So that's where you would put your eyes. There's this stage, which is where we put the smear of our sample, which I mentioned, and there are objective lenses which magnify the sample. So how would we go to look at this? We would turn it on, and we would focus on these objectives. So you want to always start on the lowest power, which is just going to get it so that we can find what we're looking for. And then as you focus at each stage, you would use these focus knobs to get the image to look clear, right? So every time that you have the image focused, you would go up to the next magnification. It goes all the way up to 1,000 times magnification. So I have a picture of that for what these germs look like. If you can tell, they kind of look like little dots, right? Those are little circles, and the way that they're forming together is in clusters. You could also have ovals or spirals, and you could have them cluster like this, where they're just kind of jumbly, or you could have them in lines. And that will tell you a little bit about the germ that you're looking at. So, now that we know the basics of lab, that concludes this video. So, we went over how to set up for a lab safely, how to take and culture a sample, and how to examine that sample. So remember this next time you're curious about this invisible world that is all around us.